Finally, Ramon Nogueira is going to present neuronal activity and its correlation with decision-making.
Okay, good afternoon, everybody. My name is Ramon Nogueira, and I'm going to speak about neuronal activity and its correlation with decision-making. First of all, I would like to introduce our scientific field, which is called computational neuroscience. And basically, it is the study of brain function in terms of information processing properties of the structures that make up the nervous system. In particular, in our lab, we are interested in understanding the process of decision-making at the neuronal level. Basically, we want to answer three main questions. What information is present in the brain during the process of decision-making? Where in the brain is this information stored? And how this information is encoded and conveyed among neurons and among brain regions? In order to answer these questions, we carried out an experiment, an auditory time interval categorization task with 3 rats. The behavioral protocol is the following. The trial starts when the rat is central nose poking, enters into the central port, then a sound is presented to the rat, and after a variable time delay, a second sound is presented to the rat again. If the time interval is short, the rat has to go to the left. Otherwise, it has to go to the right, like in this example. When the rat makes a correct response, it is rewarded with a drop of water. Otherwise, it is punished with a white noise and a timeout period. When the rat makes an incorrect response, the same stimulus is presented again, so that rats can use both sensory information and task history information to perform the task or to maximize the reward income. So in this plot, we have in the x-axis the sensory performance, and in the y-axis the history. The sensory performance is just the percentage of correct trials the rat is doing. And in history is the amount of how much the rat relies on the past history to perform the task. So once the rats understood the behavioral protocol, we started recording neurons from their orbitofrontal cortex while performing the task, of course. Orbitofrontal cortex is a region of the prefrontal cortex, and it's very well known for being involved in the process of decision-making. Neurons encode their information through their firing rates, which are the number of action potentials a neuron discharges in a particular period of time. Okay, so in order to extract this neuronal information, we used a linear classifier. In the x-axis, we have the firing rate for neuron 1, and in the y-axis, we have the firing rate for neuron 2, which are 2 example neurons. And then we have blue crosses and green crosses, which represent the trials, the firing activity in trials where the rat is either left or right, respectively. We can apply then a linear boundary, and then we will classify our following trials, our upcoming trials, depending on in which side of this boundary they fall. We define the coding performance as the percentage of correct classifications our decoder can make. So here comes the point, the interesting point. The green dot is the decoding performance of a decoder or a linear classifier trained to decode what's going to be the decision of the rat in the current trial. We used the neuronal information at the beginning of the trial, when the stimulus hasn't been presented yet. This horizontal line means the chance level, no information at all. So we see that even the stimulus hasn't been presented yet, there is information about what's going to do the rat in the following trial. If we perform the same analysis once the stimulus has been presented and the rat has made a decision, we see how the information has increased quite a lot. If instead of trying to decode the current trial decision of the rat, we want to decode the previous trial decision of the rat, we see how the dynamics of the information are completely different. In this case, it decreases over time. So if we plot in the x-axis the amount of history information or the amount of reliability the rat uses in the past against the information the rat has at the neuronal level about the upcoming choice, we see there's a clear correlation. So rats use multiple strategies to perform a task, and we can see that at the neuronal level. So finally, to wrap up, I would like to say that rats use both sensory information and task history to perform the task. We find information about previous trial decision and upcoming trial decision, and each has its own dynamics. There's a correlation between behavior and neuronal information. So finally, I wanted to thank you all for your attention, as well as all the people that contributed to this project, and of course, the organizers of the JIPIs, and thank you all.